This is your lab class number 10, and in this lab class we will be looking at PDMS molding. Now, what exactly PDMS molding is? If you remember in our theory class, what we have shown is that you have a microfluidic chip, right. So, in the microfluidic chip you should have a silicon mold, right, on which you will pour the PDMS, and when you cure it you can peel off the PDMS. So, this process from using a silicon mold and pouring the PDMS, right, and then curing it and peeling it off, this entire process will be shown to you in this particular lab class. Alakya will be taking this particular lab class which I have designed for this course, and you will be looking at very important experiment, and if you know this process, your life will become very easy when you are working on microfluidic platform, because in a way it is very easy to replicate the same kind of channels if you have a silicon mold with you, right. So, it is only one time you have to prepare a mold, and you can use it for n number of times if you are careful, alright. So, we will be teaching this particular technology, where we will be showing it to you how can you create a how can you use PDMS to create channels if you have a silicon mold. So, focus on this particular lab class, and I will see you in the next lab class. Till then, you take care. Bye.
Hello everyone, welcome to the course on sensors and actuators. In this module we will study what is PDMS molding and how does it become important when you are trying to understand how to fabricate biosensors. So, like we have seen how sensors can be fabricated using silicon wafers, the process, the fabrication process, how lithography, the choice of wafer, and the different process steps to achieve a MEMS based device. Another part of sensors are at a micro scale level are PDMS based microfluidic devices. Again, when we talk in terms of making biosensors, it could be as small as, the dimensions could be as small as a micro nano scale. So, how do we replicate this? Unlike the standard optical lithography process, where we have the photoresist and we follow the standard procedures, there is another alternative in order to fabricate very quickly a biochip. So, an important part of biochip fabrication is PDMS. A lot of chips have been fabricated using this polymer. So, PDMS stands for polydimethylsiloxane. So, it is a polymer of carbon and silicon. Let us see how we can fabricate a bio-MEMS device using this polymer. So, why do we have to fabricate using this elastomer? So, all in all, over a wide aspect of making microfluidic devices or bio-MEMS devices, the choice, most commonly opted choice is PDMS based polymer. So, why is this most opted one? When you see our fabrication point of view, it is very easy and rapidly it can be prototyped. And when we talk in terms of cost, so they are reasonably very low when you relatively compare it with the standard fabrication procedures of MEMS based devices. So, when they are available with you, you can rapidly design and then fabricate your bio-MEMS device using the polymer. Now, let us see how step by step we can achieve a microfluidic or a bio-MEMS device. Moreover, another major advantage of using this polymer is it can, once we mold it into a device, the chip would mimic the environment of a biology, like it can easily mimic the physiological, chemical and the biological environment of, for example, the human. So, the entire mechanism of the blood flow through the veins, all of this can be scaled down and replicated, like you would have heard of terminologies like lab on a chip. So, all of that is based on this means of fabrication. So, we would fabricate the entire organ. There are experiments where which is again called as organ on a chip, where the functionality of an organ has been mimicked onto these biochips, and that is why they are called biochip. And another different application is widely studied is if you want to study the antigen antibody susceptibility, the microbial test. All of this again you could fabricate a biochip and then study the antigen antibody susceptibility, antimicrobial test, or you could even do the impedance test, impedance analysis on the microorganisms. So, all of this protein interaction, so the list goes on, the applications are vast. So, when we want to start making a biochip, let us try to understand how do you the basic steps which are to be followed in order to make these devices. So, initially let us see what are the requirements. So, here what I have is, as you can see, this is a silicone elastomer, Silcard 184. So, this here is what, with this is what I am going to make the PDMS molding. So, this silicone elastomer along with curing agent. So, if you want to make a mold using PDMS, then you might consider buying this elastomer and a curing agent. So, elastomer and curing agent are mixed in certain ratio. So, this is 10:1 ratio. In this ratio, when these two are combined, it forms a polymer which can be cured, molded easily. You can replicate micro to nano level features in just few hours. So, now that we have seen how the curing agent, as you see, it is more transparent, and this you would see is more viscous. So, once I pour it into a container, you will see how the two are immiscible initially, and then after a lot of stirring, then it becomes a uniform mixture. Now, let me take the two in 10:1 ratio. So, you could use a container in order to quantify the amount of elastomer that you are taking. Here I have this handy. So, I would take 5 ml of silicone elastomer into this container. As you see, this is a highly viscous polymer, the silicone elastomer. Now that I have taken some amount of elastomer, now I take the curing agent. So, I need to take 0.5 ml of curing agent. Yeah. Now that we have, if you see, they are viscous. As you see, the curing agent would be floating on top, and then at the bottom I have the PDMS, the silicone elastomer. Now what we have to do this is, we have to stir this well. So you could take a stirrer and then stir them thoroughly, and what happens while I do the stirring is, there are a lot of bubbles which get formed. So this process would go on. In order to make the two immiscible polymers, combine them into a single liquid, so you need to stir it for around 5 to 10 minutes thoroughly, so that the curing agent and the silicone elastomer are mixed. What happens if you do not stir them well and you just use it onto your mold and then cure it? If you see, later you would see cracks which are, they become more brittle. So it is not a flexible elastomer. When you cure it, when you remove it from your mold, you will see that they are more brittle and they tend to break even before you dice them in order to get your device. So as you can see, I have stirred it for around 10 minutes and there are lot of bubbles which got formed while I do the stirring action. Again, a precautionary and important thing is, when you are doing the stirring, make sure you do not inhale them, do not keep them very close to your face, so that while you stir, there are a lot of chemicals. There are a lot of chemicals which comes out through them once you do the stirring, and so you need to keep them at a lower level, do the stirring, and then ensure you have a mask, because it is not recommended to do it with just having this in your hands and then inhaling. This could be harmful. Once we have the curing agent and the elastomer mixed thoroughly, let us see what to do with this mixture. Here what I have is a desiccator. So if you have not heard of what a desiccator is, so this is a chamber which will provide vacuum. As you can see, this is the top lid which is transparent, and here this is the bottom casing, and here there is an elastic rubber so that the lid sits tightly and then the vacuum does not. So this mostly is a vacuum chamber, a small chamber. And if you would like to know what are the applications of desiccator, they are vast. There are a lot of purpose. However, this provides a clear, neat vacuum environment inside the chamber. How do we generate this vacuum? So here you can see the chamber is connected to a pipe which goes to a pump here. The pump here is having a dial gauge. So all the air that is inside will be pulled by the pump here and a vacuum will be created inside this chamber. Another important application of using desiccators here is, say you have fabricated your device and you would like to characterize it in a facility which is farther from where you are staying. So all these devices, in order to protect them from contamination or even to prevent, like when you say silicon wafers, they are hydrophilic, they are hydrophilic in nature and they always tend to absorb the moisture in the environment. So in order to prevent, what happens when the silicon wafers, assume you have a clean wafer and you have to carry to your office or your department, and what happens is, when you carry to the environment, it is always advised to not use them, handle the wafers with bare hands, because it can cause contamination. And another thing, they are not exposed, they should not be exposed to moisture or air, because they absorb the air and the moisture which is there in the environment, and there is oxide layer which gets formed. So the silicon oxide which is formed, the native oxide, is a few nanometers. However, it can hamper the device functionality. assume you want to deposit metal which is like 1 micrometer, and then you just get your wafer, and there is already oxide which is formed on your wafer, and not taking any precautionary measures. So, there is some 2 or 3 nanometer of oxide which is there on your wafer, and then you deposit metal. If you desire to have 1 micrometer metal deposition, that is not what you are going to get, because in between there is contamination, the oxide, and that could hamper your device functionality. So, in order to avoid all such scenarios, you have to use the desiccator. In our case, when we are talking about the biomems or the MEMS based sensors, so this becomes important so that the wafers are not exposed to any sort of contamination. Now that we have the desiccator here and the vacuum pump, what I am going to do with this PDMS mixture is degas. So, degassing is done using the desiccator to remove all the air bubbles. If you see, there are a lot of air bubbles now which got formed while I was mixing the two polymers. So, in order to remove these bubbles, we do not want these bubbles directly. You cannot use this mixture for curing, because we are talking about making biomems device which have channels of a few micron length, and the dimensions are so small that even a droplet or even a bubble here can block your channel, and that is not what is desired. So, even tiniest of the bubble has to be removed from this mixture. So, we follow this procedure called degassing, where the mixture will be put into the desiccator for around 5 to 10 minutes, desiccated so that all the air bubbles will be pulled out, and inside the vacuum chamber it would ensure that there is no more air left, and this you will see it would turn to be a clear mixture, transparent clear fluid. So, now I am going to put this into the desiccator. I have my lid properly closed, and then I turn on the vacuum, the pump. So, you could see the dial gauge. If there is any leak, then the pressure would not increase. So, now I have the tube, the container where I had the elastomer is placed inside the chamber, and then the vacuum process is going on. As you can see, there is some amount of pressure, that is the vacuum is being created inside this desiccator. There, I am not sure if you can see inside how the bubbles are coming towards the top of the surface. So, there are a lot of bubbles in the beginning. So, after you have properly whisked the elastomer and the curing agent, there were a lot of bubbles which got formed, and then that removal can happen using the process of desiccation. And now this process would take around 10 minutes, and then you will see all the bubbles gone out, and then you have a clear liquid. So, this was nearly after 10 minutes of waiting. Now this is what you can see. There were a lot of small tiny bubbles which were there inside the mixture, but now there are very few bubbles. So, we will wait for a couple of minutes, say 5 minutes, and then you will be able to see all the bubbles flown out, and then inside the vacuum chamber we would get this clear polymer mixture. Having given around 15 minutes of time for desiccation, now that there are no more air bubbles, what I am going to do is, here I switch off the pump, and then you have to slowly release this valve here, because if you do this in a very fast very quickly, if this knob is turned, what happens is there is a lot of air which gushes inside the chamber, and that can again form air bubbles into the mixture. So, that is not what we want. So, what we will do is slowly degas. So, there is a slight noise, if you can see, and that is nothing but the air which will be going inside the chamber. Now, after degassing, you can remove the lid, and then here is the clear liquid. As you can see, there are no more bubbles into this polymer mixture. So, now that we have a clear mixture, like let me just repeat the process. We take in 10:1 ratio the elastomer and the curing agent, mix it thoroughly, and then once we have the mixture ready, we put them into the desiccator and desiccate it, degas for 10 to 15 minutes until you see a clear mixture like this. Now, what do we do with this mixture? So, here I have a silicon wafer. So, here this is the wafer that I am having. If you are able to see the fine lines here, so this is embossed on the surface. So, this wafer has structure like this, a pyramid like, if you see this, the rhombus structure. Here this is an embossing. So, now what we do is have this mixture, and then here on the wafer we see an embossing like this, but you could have many different forms of this structure. So, if you want micro channels in your MEMS based device, the BioMEMS device, then you could just have the micro channels engraved on this, and what would happen is, once I have this PDMS layer, I would pour it onto this mixture, and then I am having it very close to the surface, because even if it is poured at a distance, when you pour it from a very far distance, then it would tend to create bubbles on top of it. So, there is a very small amount of polymer mixture what I have taken, depending on the dimensions, depending on your device. So, you could choose what is the quantity which would be more appropriate to get the device. There are absolutely no air bubbles, as you can see, and if by chance there are any air bubbles, then what you could do is, you could remove the air bubbles using a small needle. As you can see, the slight mixture, the polymer mixture which I have poured onto the embossed surface on the silicon substrate. Another thing what you have to take care of is, while you do this process, make sure the substrate what you have is thoroughly cleaned. So, you can follow this procedure, like clean it with acetone followed by IPA and then some DI water. So, be it the silicon substrate or any other substrate on which you want to fabricate the device. So, follow the cleaning procedure and then have the PDMS liquid poured onto the device. Now that we have seen how the liquid goes into this, we have to cure this. So, the setting what we have to follow is put it in an oven at 70 degrees Celsius for 2 hours. So, what happens if you set it at higher temperature? The curing at higher temperature makes the mixture brittle and it tends to break, the device tends to break. Hence, optimum temperature is 70 degrees Celsius for 2 hours. So, what you have to do is set the oven temperature to around 70 degrees Celsius. So, take some time to increase the temperature. Now that we have the oven at 70, when you can place your mold for 2 hours and leave it here at 70 degrees Celsius. So, after curing the mixture at 70 degrees Celsius for 2 hours, you get back your mold and then you could cut it using a surgical blade, and this is what you get. So, this is the flexible PDMS substrate on which this structure was embossed. Now, if you see this, you can observe how you can easily replicate the structure on your mold onto PDMS devices, and you could even mold channels of a few micrometers and nanometers in dimensions using this polymer. And another thing what you can see here is there are tiny bubbles at the end here. As you can see, this is our device structure what we desired, but then there are few bubbles at the end. So, always make sure you remove those bubbles. What happens, so here it is still okay to have the bubbles at the end because it is not hampering the within the device dimension, but if that bubble, so what happens is when you do this sort of molding and then curing, bubble tends to retain inside the mold. So, that structure would, if that comes the bubble is somewhere here in between your device structure, it could block your channels. Ideally, we are trying to make devices with micro channels and then flow fluid at some rate, so that we are able to get droplet formation or we are able to send bacteria and then see its susceptibility. So, for all of these tests, mostly they are liquid based, and when they are flown through these channels, they could create some sort of blockage. And that is why you always have to make sure that you do not have any sort of bubbles while you do the entire process, from being mixed to being cured and all of that. The degassing becomes an important process. So, now that we have seen how you can replicate your structures or features very easily in just a few hours using PDMS, this can be a base with which you can take help and then start making bio-MEMS devices. So, when you make these biochip, once you have your chip, you can do the desired study. This was an introduction about how to do PDMS molding and how this can be integrated into making a MEMS based biochip. That is it. Thank you.